Hey, I'm delighted to be here to celebrate the 10th anniversary of PME, and particularly in immunoengineering, and tell you about our work developing microbiome modulating therapeutics. So I want to begin by asking the audience how many of you either have a food allergy or know someone with a food allergy. Yeah, virtually everyone. But when I was a child, this wasn't the case. We now have in the United States 32 million Americans suffering from food allergies. This includes both children and adults. Almost half of both of these groups experience a severe reaction. And when this is enumerated by insurance claims, we can see that between 2007 and 2016, there was a 377% increase in diagnoses of anaphylactic food reactions. So this is a very real and life-threatening public health program. People can be allergic to any food, and they can become allergic at any point in their life. But these foods are the biggest offenders, and they fall into two big groups. Milk, eggs, soy, and wheat tend to elicit reactions that are limited to childhood. Many children outgrow those allergic responses. But peanut, tree nuts, fish, and shellfish elicit lifelong, life-threatening allergic responses. And that's the problem we want to focus most on. As I mentioned, when I was in school, food allergies were rare. By the time my children graduated elementary school, and they're now 25 and 29, there were 2 children in every classroom that had an allergic response to food. Classrooms became peanut-free. So this is a generational change. How can we explain a generational change in susceptibility? So our focus has been on the microbiome. Our understanding of the microbiome has exploded during this same period of time. And we now know that microbes populate our skin and mucosal surfaces and profoundly influence our health. We're just beginning to understand them at a genetic level, not at a community level. But we do know that while our genome encodes 23,000 genes, our microbiome encodes over 1 million genes and growing. So let's just stop for a minute and let me explain to you how we define the microbiome. And it's the collective genomes of all of the microbes that live either inside or outside the human body. And includes bacteria, bacteriophage, fungus, virus, protozoa, in the developing world, protozoa and helminths. As I go through this talk, I'm going to be focused only on bacteria. That's the only group we've studied, but I may use the words bacteria and microbiome interchangeably. So why would we look to the microbiome? How could the microbiome be promoting this increase in non-communicable diseases? This slide shows food allergy. It's from a review about food allergy in the microbiome, but I could line up on the side of this slide a whole list of non-communicable diseases like obesity, inflammatory bowel disease, autism, multiple sclerosis, all of which have been associated with changes in the microbiome and all of which are rising in parallel. They all have genetic predisposition, to be sure, but that can't account for such a dramatic increase in prevalence in such a short time period. It's lifestyle factors that have changed the diversity of our microbiome that most scientists believe are driving this increase in non-communicable diseases. And these include, just briefly, overuse of antibiotics, not only prescribed antibiotics, but antibiotics present in our food and water supply. The consumption of our modern day high-fat, low-fiber diet, very different. You know, our microbes eat what we eat. Our ancestors were consuming a diet very high in fiber. As I'll show you, dietary fiber is very important for the maintenance of homeostasis in the gut. We've acquired our microbiome from the environment, and we've moved from rural settings to urban and suburban settings, even sealed buildings, where we have less opportunities for microbes to contribute to our diversity. Vaccine-enhanced immunity has reduced infectious disease. The presence of pathogens changes our microbiome. Of course, I'm not saying that vaccination is not the greatest public health success story in history, but the absence of infectious disease correlates with the rise in non-communicable diseases. And finally, we've intentionally eradicated pathogens that have coexisted with us for centuries, including intestinal helminths. All of these factors together have changed our microbial diversity, apparently in a way that's depleted protective populations of bacteria, and that's what I'm going to tell you about. So we asked, can we bioengineer microbiome modulating therapeutics for food allergy? I'm intentionally going off theme and using the word bioengineer instead of immunoengineer, and I'll explain why. When we began this project, we worked with a collaborator from Naples, and he provided us with fecal samples from healthy and cow's milk allergic infants. And you can see right away, if you look at these pie charts, that these are infants that are only 4 to 5 months old. The healthy infants had the bacterial populations we expected. Lactobacilli and bifidobacteria are your yogurt-type bacteria. You can buy these bacteria at any Whole Foods. They've been known as probiotics for many, many years. These are greatly depleted in the cow's milk allergic infants, who instead have a microbiome that looks more like what you see in adults. So for study of food allergy, we don't have access to biopsies, so we wanted to develop a really good model to test our drug candidates. And the way we decided to do that was to take feces from either 4 healthy infants from this Italian cohort or 4 cow's milk allergic infants, transfer those feces into germ-free mice, mice that have no microbes at all, sensitize those mice to a cow's milk protein, it's abbreviated as BLG, beta-lactoglobulin, plus an adjuvant, and then challenge them to look at their anaphylactic response. These experiments were led by a very talented graduate student, Taylor Feely. One trick we were able to use was, I told you that the microbiome, our bacteria eat what we eat. It's very often that people have been unsuccessful in transferring the microbiome from humans to mice. We had a trick we could use because our human subjects had a very restricted diet. They were only consuming infant formula. So we gave this exact formula the infants were consuming to the mice. And that really facilitated engraftment of the human microbiomes into the mice and helped us to create a very reliable model. And this is just a snapshot of the data that we found in this study. This graph represents 100 mice selectively colonized with either the healthy or CMA microbiota, or they remain germ-free. In mice, we measure an anaphylactic response by looking at hypothermia, a change in core body temperature. This is not a measure of food allergy in humans. And what you can see is that the mice that got the healthy infant microbiota are completely protected from an allergic response to food. The mice that got the cow's milk allergic microbiota are susceptible. The germ-free mice are susceptible. If we look at antigen-specific IgE or IgG1, accordingly, it's higher in the cow's milk allergic mice than it is in the germ-free mice or the healthy mice. The depletion of mast cell proteases correlates entirely with the induction of this anaphylactic response. Those are the mediators of anaphylaxis. So they're elevated in the mice that have the drop in core body temperature. This slide then is proof of concept that there's something in the healthy microbiota that we can discover and use to treat or prevent food allergies. So that's what we went on to do. We noted through detailed bioinformatic analysis and analysis of changes in gene expression in the healthy and CMA colonized mice that there was a particular bacteria called Anorostipes caseae that stood out as being representative of the healthy microbiota. So much so that when we transferred it by itself, it was almost able to mimic the protective effect of the healthy microbiota in preventing the drop in core body temperature and reducing antigen-specific responses to BLG and a reduction in mucosal mast cell protease 1. I mentioned to you that we produce short-chain fatty acids, namely butyrate, acetate, and propionate, by the fermentation of dietary fiber. This bug, Anorostipes casei, is a super producer of butyrate, shown here. So there are many different companies that have grown up that are exploiting our understanding of the microbiome to develop live biotherapeutics. Most of these are focused on transferring either whole fecal material or consortia of bacteria or bioengineered bacteria. But all of these bacteria are obligate anaerobes. They're very oxygen sensitive. And how to transfer them and get them from the laboratory through an environment that contains oxygen and into the patients is an enormous challenge. And it has to be solved empirically. You know, how do we dose these? What amount of bacteria do we give? For how long? For what duration? What formulation do we use? We decided to form a startup company that takes a different approach. Was company Cluster Bio, was co-founded by myself and Jeffrey Hubble. John Flavin, the former chair of the Polsky Institute for Innovation at the University of Chicago, and now the CEO of Portal Innovations, a life science and venture startup facility, is our executive chairman. And what we've done is to create a platform for targeted delivery of microbial metabolites. Our drugs have a polymeric backbone such that we can change the charge on the polymer to alter the delivery location of the metabolite. There's a linker that varies the release rate of metabolite. The polymer backbone is stable, safe, excreted intact, extensively used in industry. As I told you, our first generation metabolite will be butyrate, but we have others, discovery metabolites waiting to be developed. The formulation aggregates into my cells with a hydrophobic portion and a hydrophilic corona. So here's just a little bit of data about our drugs. We know that when we change the charge, we can have a neutrally charged micelle, and this is neutral, butyrate micelle, or a negatively charged butyrate micelle. And this simple alteration dramatically changes the pharmacodynamics of these drugs, which I'll show you here. So the neutral formulation is released in the ileum, the last part of the small intestine, and goes back to baseline, the red line, pretty quickly, within 4 hours. So we sacrificed mice at each of these time points and measured butyrate in the luminal contents. We don't see release of the neutral formulation in either the cecum or the colon. The negatively charged formulation, by contrast, is not released in the ileum, but it's released to high levels in the cecum and for a longer period of time. We don't detect its increased concentration in the colon. We think that's because it's rapidly utilized, and we also think that these high prolonged levels in the cecum provide a continuous outflow to the colon. So with these two formulations together, we can cover the whole GI tract. So we tested this in a model of food allergy. And what we've done here is created dysbiosis by treating the mice at weaning with antibiotics, continued that throughout the sensitization protocol. The sensitization is similar to what I showed you in earlier slides. Some of the mice were challenged with peanut to document a uniform induction of an allergic response in the group. That's our positive control group. Other mice went on to the therapy with either our butyrate, a mixture of our two forms of butyrate twice a day, or PBS. And what you can see very clearly here is that our butyrate formulation protects against an anaphylactic response to food. That's the green symbols, compared to the PBS control. As always, the high concentrations of mucosal mast cell protease 1 correlate with the drop in core body temperature, significantly reduced in our But-M treated mice. So is peanut-specific IgE. And this is how we think it works. The polymer backbone has butyrate bound to it. Butyrate is cleaved from the backbone in the lumen. The polymer backbone is excreted intact. Butyrate gains access to the lamina propria. It interacts with innate lymphoid cells to induce the production of IL-22. In data I don't have time to show you today, induces the secretion of antimicrobial peptides, mucus production by goblet cells, strengthens tight junction proteins. In other words, it restores the gut barrier protective response. I point out that this formulation is allergen agnostic. It does not matter what allergen you're allergic to because we're targeting gut barrier integrity. And that's why I said bioengineering instead of immunoengineering. And finally, I'd like to end by thanking the talented group of people working in the lab shown here. We're a mix of PME and BSD and our funding sources. Thank you for your time.
Unfortunately, we're kind of out of time, but I want to introduce the next speaker, who is Professor Sam Riesenfeld. If you have questions for Professor Nagler, I think you're sticking around a little while later for during the break after all the presentations. So, Professor Riesenfeld started in 2019 to work at the interface of machine learning, genomics, and systems immunology here at UChicago. Recruited her from the Broad Institute, and we're really excited about your work in this area. Recently received a Young Investigator Award from the Cancer Research Foundation, like I mentioned, and all of this work is allowing her to contribute towards identifying novel roles for rare immune cells in allergic responses and whatnot. And today, her work is imaging and transcriptomics and genomics in the field of cancer. So, thanks. Yeah.
All right, let's see. You hear me? Okay. Thank you. I'm really very happy and honored to be here celebrating with you. I realized that I absorbed the PME celebratory, emphasis so much that I woke up this morning and put on a PME color without even realizing it. As Sean said, I've been here for about 2 years, and I've really found it to be a fantastic research environment. Let's see if I can, which thing is the thing? That's the thing. There we go. So my lab does systems immunoengineering and computational immunoengineering, and that really means that we weave together statistical and machine learning techniques in order to leverage large-scale genomic data to answer questions in systems immunology, cancer. We pivot a little bit because some of the techniques apply to other areas, but we also really value, the expertise that we've gained by working in immunological areas. So in this talk, I'll highlight a project and some early results that was really motivated by the question, how can we better predict a cancer patient's response to treatment or decide which treatment to give? So you are probably aware that in the last few years, there have been a lot of different targeted cancer treatments. And they target, specific processes or cell types. And that's very different from, say, generalized therapy like, chemotherapy. So as an example, which you might already be aware of, if a lot of cells in a tumor express PD-L1, that's the ligand for PD-1, which is a checkpoint inhibitor, then that means they're evading immune response. And so by blockading PD-1, you can hopefully get the immune system to actually appropriately deal with the cancer. And it turns out that that can work spectacularly well, but also a lot of the time it doesn't work at all. So you don't get any response. And that trend is sort of true for many different kinds of therapies. Some work in more cases, and there are some really big success stories. But as a general rule, the biomarker doesn't tell us enough that we use about how well or how likely someone is to respond to the treatment. It helps, but it's not enough. So let's, right now I'm going to kind of delve into some reasons why that's the case and what we think we might be able to do about it. So let's see, I have some notes just to make sure I don't forget anything. Right, so I'm going to go back to some basic molecular biology here just to kind of emphasize that genomic technologies allow us to sample from this flow of biological information through cells. And at the top level, we've got DNA. This is a little bit of a simplified model, of course, but that's the core set of instructions. And so, of course, a lot of biomarkers represent mutations in DNA, but there's also a lot of context around that. Am I messing something up here? Okay. There are, control mechanisms that regulate those instructions, like how accessible different DNA regions are, and then the actual transcription of genes and which sets of genes get transcribed to what extent really is the key to understanding cellular identity and their activities, and that's dynamic, right? That changes in response to the environment and other, other changes internally, and then of course most genes don't actually do anything until they get translated into proteins. But it's just a little harder to use genomic technologies at this point to get a really fine resolution, although we can get some really good protein information with them. So let's see, coming back to this question about what's going wrong with biomarkers, right? One thing might just be that the mutation doesn't always drive downstream biology. So sometimes it really is influential, and sometimes there are secondary mutations that just happen because there's a lot of instability, and they're called passenger mutations, right? They're along for the ride. You probably heard about that. Of course, there could be other factors like control mechanisms, signals from the environment that influence whether a mutation has the effect you might expect. Another thing that comes up in practice, so I'm told by my clinician collaborators, is that you can get multiple biomarkers, right? These aren't mutually exclusive, and then it's not clear what treatment should be. And if we're computational, we're like, just throw everything at the patient, and that can cause a lot of toxicity, right? And sometimes even conflicting effects. So that's not always a good solution. So, it's not clear what to do in that case. And I would say in general, biomarkers don't reflect the fact that cancer is really multidimensional and complex. And, there's a mixture of phenotypes. So we can think about we can think about a coordinate axis for sort of each major kind of program or cell type, function, activity that cancer cells and their other cells, immune cells in their environment might be doing. And so we could characterize tumors by how much of that activity there is, right? And so if it's a lot, then they'll have a high value in that coordinate, right? And this is an abstract idea. It's not exactly math, but it really is very helpful to think about it that way. So some tumors will have mixed compositions, right? They might have different features that are important to different degrees. And it turns out that that's actually true at the cellular level. So not only do tumors also, have different proportions of cell types, but those cell types themselves can express programs to different degrees, right? So there's not even a clear, discrete thing that's like the cell type. And that was shown really beautifully in glioblastoma a couple of years ago by a colleague of mine, a former Brody, who identified these 4 states in glioblastoma using single-cell genomics and other analysis. And he also showed that there were different mutations associated with each of those states, but they occurred in different amounts in different tumors. So we can really think about this all points to thinking about the space as a continuous space, right? And that's even more emphasized by the fact that over time and in response to treatment, things move, right? So cells change what they're doing, tumors change composition, and so that's also kind of a continuous process, and it might be important to really understand that. And a thing that I've been thinking about recently is also that there are some states that just don't happen, right? Just because we can have every value and every coordinate doesn't mean that they actually exist together. And so that can actually teach us something too, being able to identify sort of holes, right? Things that don't happen together, like even different cell cycle states that can't co-occur, that can be informative. So, right, those programs that I showed you are abstract ideas. they're really built on sort of this notion of the hallmarks of cancer, which has been established for a while. What we want to do is use data-driven definitions of these axes. We want to find in the data, good ways to capture the variation that's actually in the patient space, because, those abstract definitions could be defined in lots of ways, depending on what data you have, and it's not even clear that those are the best features that we should be using to characterize patient samples. So just to give you an example, in RNA-seq data, right, you might have a value for every single gene for a given sample, and that's like potentially tens of thousands, certainly thousands of coordinates. But we don't need this huge vector to describe all of the patient data because genes work together. And so there's really like components of genes that do different things. Those can be overlapping, and we can characterize the full space in using this like low dimensional representation. And what we want to do is kind of learn that from the data, learn that representation. One way that we can start to learn those features is by factorizing a data matrix, which I'll talk a little bit about here. There are a lot of ways to do that. So here I'm basically giving a schematic where each color corresponds to a learned component. And generally we would learn more than 3, but it could be quite low dimensional. And those components basically are split up across lots of genes, but they represent kind of one idea, one function. And I haven't really talked about what data we're going to use. One of the things we typically use, we typically start with public data. TCGA is a really good resource because it's got lots of different modalities. But we also will talk about other kinds of data that we want to use. And one thing I haven't mentioned is the importance of images, which we'll come back to. So I just want to mention that another thing that we care a lot about is learning the actual relationships. So really, Euclidean space isn't actually usually the best space. And this might be a little too mathy, but it's nice to have the picture in your head, right? That they live in kind of a curved space. And if you draw a line from one sample to another, and it's the shortest line, that might not actually have anything to do with what's biologically possible, feasible. So we want to actually learn the space so that we can learn relationships that really happen. And that might also be important for understanding how treatment is going to push a sample in a certain direction, push a patient in a certain direction. So what do we do after we've, suppose we get all this geometry, we get everything right. One of the things that we can do is start to look for how samples localize and what characterizes them. So for example, we have actually found, at least in some cases, that you can deal with this question about carrier mutations by looking for regions where a pathway seems to be activated based on the gene expression, the RNA sequencing, and you see higher, mutation rates there. But there will always be cases where, maybe you didn't catch the mutation, right? So the mutation will be negative, or maybe the pathway doesn't look activated yet, or something like that. So it's not like 100%, but it can be helpful for sort of identifying where that mutation is really relevant. Another thing, the main challenge, right, is to find regions that help us define predictors of treatment response. That's one major goal that we have. And that's harder than just defining features, obviously. So one of the things that we do is we try really hard to focus on using different modalities. We try to use features that are supported by more than one modality. That's not necessarily the only way, but we expect that biology will be reflected across different modalities. And another thing that we're doing is using histology images. And I'll come back to that in one second. I just want to mention that in the end, we really need the right data sets also. And treatment response data that's associated with good genomic measurements is hard to come by because it's extremely, guarded. So we are working with actually partners in biotech to try to collect this data right now. And that's something we're really excited about. I think it will help take this to the next level. So given the time, I just will give you one example of how we kind of do this to make it a little more concrete. So this is a plot of the squamous cell carcinomas in TCGA based just on their transcriptional profiles. And it's a kind of a sort of manifold representation called UMAP, which is nonlinear. It's really emphasizing 2D visualization. So local things are probably right. Once you get samples far away, distances get very distorted. But it can give you a feel of how things are related. And so we basically factorize the data matrix, and then we can use the weights for any component, and we can kind of look and see, do weights tend to localize on that UMAP. Do they mean something? You know, we see one that's keratinization. That's not something that is actually usually marked by a pathologist or by cancer genomicists, but you know, it's quite apparent. We also see component that is interesting because it spans several different types of cancers, several different types of patient cancers, and it has some immune-related genes, but it doesn't look like a standard sort of, you know, PD-L1 type thing. So we're interested in kind of pursuing those more. So if you've ever seen, you know, anyone talk about deep learning, that really there's a standard pipeline. In tissue, the only thing is that you take the slide, the histology slide, and you tend to break it up into lots of overlapping little tiles to basically help the current deep learning networks to learn better from them. But in the end, you're giving each of those tiles a positive or negative label that comes from the patient or comes from the genomic analysis. So you can train the network on that, and then you can predict and say, you know, you can give unlabeled tissue samples, tissue slides, and say, tell me if you think this patient has this characteristic or not. And that's already been used to say whether or not a tissue is cancerous very well. It's also been used recently to actually detect genomic instability, which you might not think actually shows up at the level of the tissue histology. And so we're interested in which kinds of features we detect that can be actually seen in the tissue architecture, because maybe those are more relevant for you know, clinical predictions, and also hopefully they are more robust features. So I won't go too much into this except to say that some of these we can predict really well. So we binarize our weights, and we can actually predict features really well that you wouldn't really expect. You know, and you can actually kind of go in and try to understand what are the actual differences in the tissue at the level of, you know, these slides to try to see if you can learn something new, and that's still ongoing work to figure out how to do that. And then, you know, if we do all of our components, some of them look terrible, and we're actually fine with that, because you know, that's probably a technical artifact that we don't expect to actually show up in tissue slides. We expect it to show up in genomic data. Or there are sometimes programs that are represented in lots and lots of kinds of cancers, so they won't be specific to one kind. But we do learn, for example, the immune component with far better than random prediction, but we just don't know yet what it means exactly. So that's part of the work in progress. So I'll just kind of put this up there to say that one of the things we're looking for, you know, we're working on are better machine learning methods, and in particular, the Center for Data and Computing has given us a grant to work with some awesome collaborators, really across institutions, Argonne, computer science, and we're trying to figure out exactly how to do that, what students to get, but it's really fun to try to make that work. Like I said, we're really excited about getting the right kind of data. And in the end, we really care about translating this to patient care, making the kind of predictions we make eventually accessible. And for me, I'd love to see one day, you know, this turn into a collaboration on that leads to interventions or better diagnostics once we understand more. So just a quick acknowledgement. Most of this, I have this amazing group that happened in like 2 years. Hannah is a second-year student who convinced me to do this kind of work with an awesome collaborator who's a statistician and an oncologist here before she was even a first-year student. I didn't work on cancer before that. So this grew, it's very led by students, which is, I think, a wonderful thing about U Chicago. The students are just awesome. And that's it. Thank you.
All right, thanks, Sam. Actually, interest time. We got to move on to the next speaker. But yeah, stick around for questions after. We got a 15-minute break. Our final speaker for this segment is Professor Savas Tay. Give him a round of applause because he recently got tenure. Yay! And because I don't want to take away from too much more of his time, we'll just pass it directly on to him.
Okay. Thank you for the invitation. It's great to be here with real people, not just Zoom. And so I will talk about two things that has been the centerpiece of the last two talks. We have heard some really interesting topics this morning. They are about immunity, they are about cells, and there are measurements in them, right? So in my group, what we are doing is we are developing new measurement technologies that enable some of these data generation efforts. And we're also using them to understand how individual cells might be working. So at the core of immunity, of course, what you have is individual cells like T cells, B cells, dendritic cells, cells from the environment in tissues talking to each other by exchanging chemical signals. So this is a very interesting problem in itself, and it is not much different than other communication problems in engineering. So such as in electrical or optical engineering, there are a lot of analogies that one can draw. So a little background on myself. I'm actually a person who's trained in physics and engineering. I moved to doing biological research rather late in my career because I truly believe that the next century, unlike the previous two, like it's been the century of physics and then chemistry maybe, this is going to be the century of biology and medicine. That I truly believe in that. I'm very motivated with that myself. So some of the questions we try to understand using these approaches, either from engineering, experimental biology, and computation are, we try to understand how living systems organize and communicate with each other. So if you're dealing with single cells, how do single cells talk to each other with these chemical signals, deal with environmental threats? And then we ask the question, can we develop predictive computational models of these systems? After all, if you have a computational model, you can do in silico experiments much faster than you could do with actual experiments, screen a lot of conditions, and then that could really guide you in doing new experiments with actual experiments to really narrow down the phenomena that you're studying. And finally, can we manipulate complex biological systems like collection of cells, maybe a signaling pathway in a cell, or even tissues to fight disease? So these are some questions that we're motivated by. We use a combined engineering, experimental, and computational approach. And because of all that, you need to develop several technologies. So studying individual cells, a mammalian cell is like a 10 micron diameter sort of sphere. It is very small, so you need to go to the same scale of length in order to study that efficiently, and that requires some microfabrication techniques to be developed, for example. So we are in the business of doing that as well. So at the core of all these interesting biological phenomena, you have this communication problem, right? You have a cell that is sending a signal. It could be a chemical signal that goes up and down in concentration, maybe a protein, that is also disturbed by the environment. So you have this nice signal that is, perturbed by the tissue environment, then goes to a receiver cell, maybe a macrophage in the environment, and then that receiver cell interprets the signal, changes its gene expression profiles, does something. Maybe it's going to attack the invading microbes and whatnot, right? So all of these things can be studied under a microscope if you have the right control of length scales, if you have the right control of time scales, and you can study these things and even like computationally model that. So that's the type of research that we are trying to do in our laboratory. So of course, in traditional biology and also in this area, which you might call signaling or systems analysis, everything is typically done by traditional pipette and dish techniques. So you have a well plate, you have a pipette, multi-channel pipette, and a technician or a grad student or a postdoc constantly pipetting things in and out. This is very tedious, this is very labor intensive, takes a long time, and it is also not very reproducible because it's prone to human error. So what we are trying to do is really automate all this. The entire pipeline here can be made into a small device where the pipetting is done by the device itself in an automated way, in a predetermined manner, and with very small scales so that the precision of pipetting can be much more accurate. So instead of microliters, we're talking about nanoliters kind of accuracy. So you can realize these, what you would call lab-on-a-chip systems, microfluidic devices that does all this. And then finally, they don't look much different than electronic systems, because in the electronic systems like you have in your computers or cell phones, you have electrons flowing around. There are small transistors that direct the flow of electrons. Just like that, in microfluidic systems, we have small valves that direct the flow of fluids and cells. So we can basically borrow ideas and techniques from integrated electronics and apply them to fluidics and realize really accurate and high-throughput biological measurements. And in the end, you just put everything under computer control, realize these fully automated systems, even like autonomous systems ultimately, to really expedite this process. So here's an example of a device. This is a microfluidic cell and organoid culture device that we developed in our laboratory. You have these little specks here. These are all small culture chambers. We take cells from tissue. We put them in this device. There are 1,500 such culture chambers here. You can see them little specks like pixel-looking things. And all these tubings are directing the fluid flow into these small chambers. For example, you're putting small cells or organoids like small collections of cells that mimic actual tissue. And then you can deliver different drugs or signaling molecules to these systems in a predetermined manner, fully automated fashion, and then monitor what's going on under a microscope. So this is that system operating. As you can see, we are loading different reagents into different chambers. There's some mixing going on in this area. This is a little bit sped up, but it's not much different than real time. And you can essentially establish or realize like 1,500 experiments in a fully automated fashion under a microscope while the cells are being maintained and kept alive, right? So these kind of devices really expedite and make traditional biological measurements better in order to do more measurements that you couldn't do without these tools. So how does this work? Typically, we start from a biological problem we want to solve, right? Say, I want to take macrophages, activate them with different signaling molecules, measure their gene expression response, whatever it is. So you start from that problem, you do some calculations, and then you go to 2D or 3D design software like AutoCAD, lay out the devices you want to make. Then we go to our nice clean room, the Pritzker Nanofabrication facility at the PME. We make the master molds to make these devices. Then we print out these chips using some specialty techniques. And now we go and integrate them with microscopy automation, computer control, and automated readouts. So this entire pipeline we developed, and we use that constantly in our research. So there are devices to measure cell-secreted cytokines, signaling molecules from cells. There are devices for measuring different proteins with extreme accuracy in a single molecule level. There are devices for high-throughput culture and of either single cells or organoids. So depending on the problem, you change the design parameters and realize these devices, and it's a well-developed process. So here's an example of something we haven't yet published. This is a tool that takes a bunch of drugs or signaling molecules, make combinations of them, and then distributes them into small nanoliter droplets. Each droplet here is about 100 microns in diameter. So you can put cells in there, you could put bacteria, you could put signaling molecules, drugs, and this is slowed down significantly. So you can generate a single reaction and a single droplet 100 times a second. So within a few hours, you can generate 1 million reactions under 100,000 screening conditions and really expedite drug screening, for example. Okay? So these are some of the tools that we are already developing and using in our research. So what we did for this, using these tools in the past, is my group has really tried to understand the functioning of these complex signaling pathways like NF-kappa B. So whenever you're talking about immunity, when you're talking about response to an external threat like bacteria or viruses, this is one of the pathways that will be activated by the responding cells that is going to regulate the type of response the cells will mount to these external threats. So it's going to make choices about what to activate, what kind of pathogen I have, what kind of dose abundance I have in the environment and timing. Okay? So we do that, and we collect these single cell traces from live cells under microscopy conditions using microfluidics and generate a lot of data. Another thing we recently did is using our single cell approaches. Of course, once COVID hit, we wanted to help out a little bit. So a very talented postdoc near Dryman he's a virologist, trained virologist. He took these single cell approaches and live cell microscopy. He took a collection of 1,500 FDA-approved drugs and then tested them on OC43, which is common cold virus, and then 50 or so of these drugs actually work to inhibit this virus really well. And then OC43 is a close relative of SARS-CoV-2, the pathogen for COVID-19. Then we took these, went into BSL-3 environment, and then tested them on the actual SARS-CoV-2. And we found that there are about 20 drugs that really efficiently inhibited this virus in live cells. One of them is called masitinib. It's a drug that is originally developed as a kinase inhibitor for cancer, treatment of cancer. We found through crystallography that this binds to the main active site of the main protease of the virus, 3CL-Pro, and inhibits it. And further experiments with mice show that the drug is very efficient in reducing the viral loads in both the lungs and the nose of the mice. So using these tools, we were able to develop, I would say, discover that this existing drug, which already has some clinical safety profiles figured out, that actually works really well, at least in the cells and in animals, and clinical trials are to be done in this direction. So we have also used these tools for looking into different phenomena in developmental biology. We were able to generate a lot of interesting data sets by basically combining our microfluidic approaches and organoid imaging and tracking to understand how the neural stem cells in the brain during early development differentiate into different cell types and cell lineages. So the tools are really good for looking into that kind of effects as well. Brooke Schuster, who's a grad student who's graduating, did this really interesting work where we collect pancreatic cancer specimens from patients and then grow organoids out of these. And then she developed this tool where we can do different drug regimens, different drug treatments that are both temporally and combinatorially modulated. And then looked into the response of these organoids. We can see that, here in these videos, we can track these organoids automatically, collect data from them, look into the drug treatments or drug combinations that work for that particular patient rather than using a one-size-fits-all kind of an approach. This is a tool useful for personalized therapy. Recently, we have done some interesting work in sepsis, which is actually a very important problem for the entire world, including the industrialized world. And usually in COVID-19, in the severe case, what you're getting is viral sepsis, very similar to viral sepsis. So it's very relevant to what's happening in the pandemic. So we use some of these molecular markers to be able to predict sepsis outcomes in patients who will die, who will survive, and who will be discharged from the hospital really early on. And we've done some work on microbiome as well. So these tools are really useful for analyzing individual microbes, their interactions, and drug treatments under these conditions. So Bill Watterson has done some interesting work using microfluidics and human microbiome. And we also spun out a small company before COVID, of course, with Jack Gilbert and myself for doing in-home analysis, longitudinal analysis of microbiome in an automated way because nobody wants to deal with this themselves. So we designed a machine that will do that for actual patients.
I also want to circle back and see if there's any questions for Professor Nagler or for Professor Riesenfeld from the earlier presentations. Nicola?
Thank you. I have a question for Kathy and Sam, if that's okay. So for Kathy, I hope that the mice were not too picky with formula, as some little humans have known. But I had a question about the mice cells. Sorry if I missed, but you said that they were captured very well at the ileum and the cecum part. And why is that needed? Could they be uptaken in any one area of the gut and that would suffice?
So they whole GI tract, but because of either the presence or absence of negative charge, they release selectively in either the ileum or the cecum and presumably deliver to the large intestine. That's a huge benefit to us because food allergy is primarily a disease of the small intestine. Inflammatory bowel disease is a disease of large intestine. So now we can take butyrate and place it at its site of action. So butyrate as a sodium salt stinks, tastes terrible, and barely gets past the mouth. So this is the first technology that allows you to deliver butyrate precisely to the site of action at high concentration.
Got it. Thank you very much. Is it okay if we ask 2 questions?
Yeah, go ahead.
Sorry. And so for Sam, so when you make the tiles of the different tissue for deep learning input, do the tiles overlap with one another, like at random, or do you specify them and then? Shall I take it? Oh, sorry.
Okay, so I'll be honest that a lot of the deep learning, the image-based pipeline was developed in Alex's lab. And so what we did was sort of figure out how to do the genomics, like add the genomics into that. But as far as I know, it's tessellated in a very kind of like a convolutional type way, like it's just going across. I don't think it's random. I think they know what the overlaps are, but I don't know if that's actually used anywhere. So yeah, I actually have plenty of questions on how it all works over there, but I haven't bridged that side. Yeah, I mean, it also might impact training, right? Because you've got lots of samples that are from the same, and I think they do that partially because they need to kind of learn enough, they need enough input, but I have questions about that too, so yeah.
Sorry, we should probably go into break, but all right.
Maybe just one general interest question for Kathy, since it's a general interest session. But you talked about the importance of fiber and then metabolites produced from fermentation of fiber. But what can be learned from animals that don't eat any fiber at all? Carnivores or, I don't know what a hummingbird eats, just know it eats a lot of sugar. But are there learnings about the microbiome and then metabolic issues arriving from that?
So I don't know. I know that every multicellular organism that's been studied to date has an associated commensal microbiome. I don't know information about whether or not they consume fiber is clearly going to impact the functional activity of their microbiome. So probably they have some kind of a compensatory mechanism to, for example, one of the main functions of short-chain fatty acids is to provide an energy source for the epithelium itself. So that must be compensated for by some other metabolite in animals that have no source of fiber. But I don't know what that metabolite would be.
Thanks.